I want all of you to just try to imagine, though, something a little bit strange. I want you to all imagine that you are alien archaeologists. So you come here from a different planet, okay? It's the year 2300. You're not in Pingree anymore. You're in some like post-apocalyptic version of planet Earth. That's what an alien scientist looks like. So it's like an alien with a lab coat, basically. And you've come to planet Earth to study baseball, which was a sport played by the ancient humans, right? That's what your alien thesis advisor asked you to do your thesis on. So, you know, being a good alien graduate student, you do what you're supposed to do, you collect some data. And what you find are 3 artifacts that you think might help you to understand baseball. So here's the first artifact. Okay, so who is this baseball player? Yeah, Joe DiMaggio, right. So you see, you get this picture of Joe DiMaggio, right? And you think just looking at this picture, it's pretty clear what baseball is all about, right? There's a guy, and he seems to be like, he hit something, and there are these 3 humans, and they're all staring at something in the distance. Like, seems pretty straightforward at this point. So not a big deal. Then it gets a little bit more confusing, because the second artifact that you find looks totally different. Here's the second artifact, right? And we all know who this is, right? Mariano Rivera. Notice that these are both Yankees players. I don't think there's any controversy that the Yankees are the best team, but different players. Okay. All right. So now this is confusing, right? This is a second picture, but it's totally different from the first picture, right? It's just a guy. He's kind of grimacing a little bit. He has a ball in his hand. Like, whoa, what's going on here, right? It's like not good. Your thesis advisor is getting a little confused. You know, you're getting a little confused. Okay. So now here's the problem, right? You find this third picture, and your brain just completely explodes because this is the third picture, right? And this is just like, well, what is going on? There are all these humans. They're standing up. There's a big flag on the field. I don't see a batter. I don't see a pitcher. I don't see an umpire, right? So what's the problem here? If you're a scientist, what's missing in the blue shirt here? Or yeah. What's missing? What's the problem?
That you don't have a concept of that there's different players in the field that have different jobs.
The different jobs.
How they function as a unit.
How they function as a unit. Okay. Yeah. I think the second thing that you said is very accurate. So what could help you to understand how these different players, because you can see the different players, right? So I disagree with the first part of what you said. You can see the pitcher, you can see the batter, you can see, everyone standing, but how they function, that's what's hard to see, right? So, and does anyone else have any other ideas? What could help you to understand how these things are functioning together? Yeah.
The steps between.
The steps between.
Picture.
That's exactly right. And is there like a word for that? Like something like a bunch of pictures? Sequence, a movie, a video, exactly right. So now, yeah, so if you see this, totally makes sense, right? Now all the pieces come together, right? Well, this is a great play, like Vladimir Guerrero just like nails him at the plate. But it's another story, yeah. But right, it kind of makes sense all of a sudden because when they show you the replay, you don't just see an isolated batter and an isolated pitcher and isolated fans. You see how all the pieces interact with each other. It's a pitcher throwing to a batter, the batter hits something, and then all the fans like react to that. So it's a movie that's tying together these images. So now we're going to leave the alien behind. He found this movie. He went on, became an alien professor of archaeology to a very happy life. But the point I'm trying to make now is, as we segue into the paper, is that biologists who study proteins have exactly the same problem as this alien archaeologist. And what I mean by that is they can take pictures of proteins, but it's hard for them to understand what those proteins are doing just from the pictures that they take. So what is it? This is a picture of a protein. What's this protein doing? Blue shirt here in front. What's this protein doing? Yeah.
Oh, okay.
Blue is my favorite color this morning.
What's this protein doing? Okay, so...
I'll give you a hint. It vaguely has something to do with the article I passed around. But it's okay.
So, this protein is currently folding, or is it
Folding?
moving around? I don't know.
Looks like it's moving. I don't know. That's the point. Yeah, it's kind of a trick question. I'm sorry to put you on the spot. Yeah, I don't know. You're right. Yeah. Okay. Well, does anyone else want to take a guess? Does anyone happen to know off the top of their head? Orange, maroon, salmon shirt, right there. No, yeah, salmon shirt. Yeah. 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 Well, I don't know either, right? So it's okay. We're all on the same page. Yeah, and that's the problem, right? Like, what does this protein do? What's the important part? What's the functional part of it? What part moves? What part doesn't? I have no idea when you look. I mean, it's a beautiful picture. I think it's a beautiful picture. And you're lucky to have a teacher here who told me she worked on X-ray crystallography. So she took snapshots of proteins that look like this. It's a lot of work, and it's very beautiful. But without a movie, it's really hard to understand what this thing is doing. So we're going to dive into that. We're going to talk about how we make movies of proteins. First, though, I just want to take one step back and just make sure we're all on the same page. What is a protein? Does anyone here want to tell me? Blue, here. Yeah. What's a protein?
It's
If you've taken biology, right? I don't know. Yeah. It's a type of molecule, and it's
used for building your body structures.
yeah, and I'm going to show you that in a minute. But also, what makes it different from other molecules is it's a special kind of molecule, right? It's built out of a sequence of repeated units, so amino acids, right? And these amino acids get stitched together like beads on a string. And then what happens is these amino acids then form something called secondary structure. So parts of the protein kind of fold up into a stable shape, which is usually like an alpha helix or a beta sheet. And then these collections of alpha helices and beta sheets form into a tertiary structure, which is the actual functional form of the protein. What's amazing here, and what has captured the attention of many scientists, is that the three-dimensional structure of a protein and its function, therefore, is actually encoded by a linear sequence of amino acids. I think that's pretty amazing that just a collection of molecules like that, a collection of atoms like that, just know how to fold magically due to the laws of chemistry and physics, but that's a topic for another time. Okay, so protein, right, it's a special polymer, three-dimensional structure, helps it to function. And what's particularly interesting about these proteins is what was your name in the blue? Jack? What Jack said was completely right. There's a lot of them, and they do really, really important things. So this is a slide showing you just a sample of the different proteins that exist that scientists have taken pictures of. And what you can see is that they pretty much do everything important in the cell, although there are other things like, so these proteins over here, let's see, these proteins over here are involved in protein synthesis. These are involved in DNA. What do these do over here? These are involved in energy production, storage, all kinds of stuff. These are my favorite proteins over here that are involved in transporting ions across the cell membrane. Does anybody have a favorite protein? Huh? ATP. Yeah, it's here. Yeah, wait, it's over here. Wait, which one is it? It's enormous too, by the way. It's in energy production. I think it's this. I think this is.
It's down.
Which one?
One more. Yeah, right there.
This one.
Yeah.
Yeah, it's huge. It's a monster. Look at that thing. And that's like the Godzilla of proteins. Yeah.
Do you guys remember the little walkie guy?
Nigel.
Nigel.
Oh, his name?
It's in the, Nigel's in the middle, right there, the red one.
This is Nigel.
Not
Right.
just the red one, it's the red.
Can someone go up and show me the results?
It's Nigel.
This one's Nigel.
Hey Nigel. Oh, this is Nigel. Yeah. Oh yeah. Okay.
Just clarification, that's myosin.
Yeah. Name Nigel. Myosin name Nigel. Nigel. Okay. How did you guys come up with Nigel?
Bio class last year. Yeah.
It seems Nigel-ish to me too. Yeah. It's a fair thing to say. Yeah, that's reasonable. But yeah, that's what's amazing about these things, right? They, in addition to being really cool, they also have cool names. But they do everything that's important in the cell, pretty much. So the problem is now we want to understand how can we watch these proteins move to actually understand what they do. So does anyone have any ideas about how you can make a movie of a protein? That's exactly right, and that's exactly right. How much you do that? I like how you specified in close succession, and that is totally important and the key to the whole thing. But like, how might you do that? Any thoughts? Because you're totally on the right track.
Isn't that what Anton does?
Yeah.
Yes, that's exactly what Anton. So you know about Anton.
Yeah.
Okay, right. So So Anton is a machine that does that. I'm going to show you a picture of Anton in a minute. How are we doing with time?
Yeah, fine.
We're good? Okay. I'm going to show you a picture of Anton in a minute. But it's kind of a loaded question. Anyone else have any other ideas? How would you make a movie of it's going to use some chemistry, it's going to use some physics, it's going to use some computers. Yeah. Yeah, it sounds good to me. Yeah, that's exactly right. And that's what you do. And if you had had that idea 25 years ago, you could have won a Nobel Prize. Yeah. Almost, yeah. So 3 very smart guys, Martin Karplus at Harvard, Mike Levitt at Stanford, and R.E.A. Warshall, who is at USC, University of Southern California, came up with this idea. And they won the Nobel Prize 2 years ago for developing multi-scale models of complex chemical systems. What was your name in the room? Ellen? Yeah. So this is exactly what Ellen said. When they say development of multi-scale models for complex chemical systems, and what was your
Liz.
Liz? Ellen, Liz. What Ellen and Liz said is what these guys won the Nobel Prize for. They came up with a way to simulate all those atoms and make them move according to the laws of chemistry and physics using a computer. And to show you kind of what that means, just to give you a little bit of a taste of that, here's what that actually looks like in practice, right? So the technique is called molecular dynamics. That's the actual simulation technique. And it uses Newton's law, right? So who's taking physics here? Yeah. Okay, your hand went up first. So you know what's Newton's law about? What's your name?
Sophia.
Sophia. Okay, Sophia.
Well, what do you mean? What's the back?
Like, what is it? What is it useful for, Newton's law?
Which one?
Huh?
Which one?
For F equals NA. Like, what is it telling us? Like,
I don't know.
what is it telling you? What is it telling you? Not sure. Okay, like what is the mass, for example? Like, is that? No, I'm missing you.
I don't know what you're asking.
You know what I'm asking? Okay, sorry. I don't mean to like leave the witness. What I mean is that it was originally used like to understand things like how planets move relative to each other, based on the forces that they exert on each other, right? Those forces dictate how planets move, right? So what these guys, Karplus and Warshall and Levitt, realized was you could take the same idea, but instead of talking about the masses of planets and the forces on them, you could talk about the masses of atoms and the forces on those atoms. And you could use that to make a movie, an animation, of how not planets would move, but how atoms in a protein would move. Are there any questions at this point? Because this is kind of the key before we jump in. Yeah.
How are they measuring something like the mass of an atom and the force of an atom in this kind of to get how fast it's moving?
Oh, that's a very good question. Yeah. So the masses are kind of known already,
Okay.
but you could argue that one of the Achilles heels of this, you've kind of identified one of the weaknesses of this technique actually, which is that it's very, very hard to measure the forces between two atoms like that. There are some sophisticated techniques you can use. So this is why it's a model, and some biologists don't believe it, because they would say, well, I don't know if these forces are accurate or not. So a lot of work has went into getting these simulations to agree with as much experimental data as possible, like vibrational spectroscopic data. That's a good question. The answer is no one really knows. So people just like cross their fingers, throw some salt over their shoulder, hope that's right. Luke, you have a question? Yeah. Oh, no, it's 10 minutes. Oh, okay, so we're getting there. Right, so on the right, on the right here, this was, let me just play it again. This is a movie, right? So in black was the crystal structure. This is like what the actual picture of this trypsin with benzamidine looks like. And orange is the movie that we made with I made with molecular dynamic simulation of this protein with this molecule. So does anyone see anything interesting about how this molecule, this is an inhibitor of the protein. Is the movie telling us anything useful that we couldn't understand from the picture?
Could you point out where the protein is?
Oh, I'm sorry, that's not clear. This is the protein here. This is the protein, right? This is the enzyme here. And right here, this ring and these atoms here, this is an inhibitor that binds to this protein to prevent it from functioning. So I'm curious, do you guys see anything? Is this movie telling us anything that we couldn't understand just from the picture? Yeah.
That it looks like it's able to twist and change its shape and bind those three binding sites in order to protein.
Exactly. And so let's take that one step further. If you were working for a pharma company or as a drug designer, like how much you use that information, if at all?
Use that information because it's moving, it's changing its binding sites on the protein, which means the protein itself is getting slightly older.
Exactly. Yeah, exactly right. So if you wanted to make this molecule bind better to the protein, right, maybe that's your job, then what you might do is you might take advantage of the fact that in the movie you see this ring start to twist out of the plane and add functional groups here and carbonyls or whatever to take advantage of interactions with the protein that you wouldn't ordinarily see in the static picture. So that's in practice how these things get used. Okay, so I think we're just gonna probably, oh, and then I promised you, yeah, who said computers? Somebody said we need big computers to do this. Okay, yeah, Anton, who said Anton? Oh, you knew about Anton already, yeah. So this is a picture of Anton, right? This is the computer that actually gets used to run these simulations. This is from a company based in New York that really believes in molecular dynamics simulations, and they built a whole company around specialized supercomputing architectures that only perform molecular dynamics simulation. The computer's name is Anton, or Anton von Leeuwenhoek, who was a scientist who invented the microscope. So in the same way that they're hoping these computers will be used as a computational microscope to study proteins and stuff. Okay, so I think with that we can just dive into the paper. We'll go through it pretty quickly. Did anyone read it? What did you think? Well, who liked the paper? Did anyone like it? No, well that's not a very enthusiastic endorsement of my research. You liked the paper? Okay. Who didn't like it? Well, everyone else didn't like the paper, so explain why. Why didn't you like the paper?
I didn't read it.
You didn't read it, okay. Oh, I was just assuming no one else liked the paper. All right. Did anyone not like it? Because there are problems with it. No? Okay. All right, well then I'll take you guys through it then. So the idea here was to use molecular dynamics to simulate voltage gating in potassium channels. So we basically want to understand how a potassium channel works using this simulation technique. And just to refresh your mind, in this landscape of proteins, we're here. This is a potassium channel. So this was what was his name again?
Nigel.
This is Nigel. Do you guys want to give the potassium channel a name? What does he look like, you guys? Or
Kenneth.
she? What does she look like? Any names? Huh?
Kenneth.
Larry.
Carrie?
It's like a lobster. Larry.
Larry the lobster. Okay, this is, yeah, all right, so we have
Oh,
Nigel.
see it, yeah.
Larry, okay. Okay, so this is Larry the potassium channel here. And we want to understand how does he do what he does. So again, this is a static picture of Larry. You can think of this potassium channel as kind of like batteries, molecular batteries. They basically help sustain energy difference across the cell membrane. And the name potassium gives a hint to what it actually does. It's a potassium channel, meaning it conducts potassium ions across the cell membrane. But how? I mean, does anyone know? Anyone think how it might do that? Say it.
It's a concentration gradient. So
Exactly.
when you transfer the potassium across the channel, it creates energy almost.
Yeah, that's right, down the energy. But can you see the problem, right? There's not just potassium in the cell, there's also calcium and sodium and other ions. So how do you, like the cell, when it was evolving, had this real problem. How do you make sure you only get potassium ions across the cell membrane and not calcium ions and not sodium ions, right? Yeah, I don't know either. It's kind of a hard problem, right? So the way that evolution solved this problem, I think, is beautiful and kind of ingenious. And what we showed in our movies is this is a close-up of the conduction of potassium ions. This is a part of the protein here called the selectivity filter. And here in the circles are potassium ions. And here are little water molecules between the potassium ions. So first you can see something really beautiful, which is that the size of this hole in the protein, which is right here, is exactly the width of a potassium ion, which is, to my mind, amazing. There's no way that humans can engineer something with that precision, no matter how hard we try. We don't even come close. But how do you, when you watch this movie, what do you actually see about the potassium ions moving through? Any observations? Yeah.
They're being forced through by
Oh, that's exactly right. Yeah. Okay, so you got it in one. Okay. Good job. Yeah. And specifically, it's a little bit like as if you were playing a game of pool, right? These ions here have to sit and wait for another ion to come through, to push through. They don't just move continuously, right? Here, they're just waiting, and these potassium ions sit until an ion from below comes in and knocks them through the filter. Exactly like you were playing pool, basically. So that was a hypothesis people had for about 70 years, but couldn't actually experimentally test. And these movies revealed what that mechanism actually was, the knock-on mechanism. So the second question was, how does the potassium channel open and close? That was the second question we were interested in. And here is the potassium channel, and you can see all the action, right, is happening here and here. This part is pretty static. This is where the potassium ions move. But there are these two other domains of the protein are extremely flexible. Again, you never see that picture of a protein. And I don't know if it's completely clear, but I think what you can see when the movie will replay, just bear in mind where these are located now. They're kind of, well, what do people see? Because they haven't seen it. Well, let's wait till look at where they are now and then look where they are now. Does anyone see the difference? Yeah.
Can you replay that?
Yeah, I can't make it play faster than that. Give it one more second. Yeah, let's look replay on this one. Well, anyway, basically these parts of the protein are detaching and they're yanking down on the protein as a way to close it up here to prevent ions moving through. So it's like a lever sort of that you kind of pull down. Yeah, there it goes. Okay, so that helped us to answer the question of how potassium channel opens and closes. I think at this point you'd be justified in saying, well, this is cool, but like, why should I actually care about this? Like scientifically or biomedically, like, is this actually useful, or are you just like a frustrated Steven Spielberg or something like that? Right? And the answer is a little bit of both, but they can be very useful if you have mutations in a protein that cause a disease, like cardiac arrhythmia, and you want to understand at an atomic basis how is that happening so we can develop a drug to fix it. And that's exactly what we did in the third simulation. This is a part of the channel that does not usually conduct ions. Usually nothing goes through this part. But you can see in yellow, these ions start to travel through if you have a specific mutation here that some people with cardiac arrhythmias have. So it's as if you opened up a hole in your membrane and you have this additional flow of ions through it that can totally destabilize the cell membrane and lead to these cardiac arrhythmias, these irregular currents through the cell. So this was called the omega current. It was the first demonstration of how this could be happening at an atomic level. And you can imagine that one way to cure this might be to develop drugs to try to prevent these ions from coming through the membrane, which some people are working on. Yeah, 5 minutes? Okay, we're almost done. All right, so that's it for the movie pretty much. And also, if you're involved in, that's essentially the highlights of this paper. Did anyone else have anything about the paper that stood out to them? Did anyone else actually read it? Luke read it.
I read it.
All right, well then we can talk about it later. All right, so if you like these movies that I showed you, you can get involved in molecular simulation as well. We know people who work in it that it's not an easy field to get involved with. There's like lots of big computers and a lot of coding and stuff like that. So we developed an easy way to get involved, a game version of it basically, called Folded. and it's a computer game you can use to discover the shape of proteins. Thousands of people play this now around the world, and if you're interested in playing it, tell Luke or me about it. So it's basically, you can do everything I showed you, but interactively in a game form. All right, so that's pretty much it. If you have any questions, feel free to get in touch. And if you want to take a look at Foldit, here's the website, let me know. All right, thanks for all your time.
Thank you for
Thank
presenting.
you. Yeah.
I had passed on my phone and tried to get everyone's email. If you didn't get my phone to put your email down, can you please send me an email? We will be reinstating the Journal Club email blast shortly. So that means every Monday you guys will get an email with some stuff about upcoming journal clubs and some papers and some materials that you can look at before.